Hi, hope you're doing well. My name is Saptarshi Pine, and today we are going to talk about this poster titled Network-Based Data Integration and Visualization Provides a Global Understanding of Regulatory Mechanisms in Aspergillus fumigatus. Aspergillus fumigatus is a fungal pathogen which causes a disease known as invasive aspergillosis, which is the most common invasive fungal infection in immunosuppressed individuals. It has also been recognized as a comorbidity of COVID-19. However, treatment of this disease and combating this fungus has been difficult due to the lack of understanding of its gene regulation. Now, there has been studies which studied small groups of genes in isolation, but there were not sufficient studies for studying the gene regulatory mechanisms at a global or genome scale. To bridge this gap, in this study, we inferred a genome scale gene regulatory network of Aspergillus fumigatus and then analyzed it to come up with some solid biological hypothesis. To infer the network, we implemented a novel network inference algorithm known as Merlin PTFA, which is an extension to our previously proposed algorithm known as Merlin P. The new part is the TFA, which stands for the transcription factor activity estimation, where instead of using the gene expression of the candidate regulator, we use this gene expression, observed gene expression, to estimate the transcription factor level activity of the candidate regulators and then compare it with the observed gene expression of the target gene to come up with the inferred ages. And when we implemented this algorithm, Merlin PTFA, we saw that it was able to outperform an array of widely used algorithms such as Gini3 and Inferlater. And using this algorithm, we applied it on to a collection of 20 bulk RNA-seq data sets consisting of around 10,000 genes and 300 samples. We also initialized the network with the prior network based on TF binding site motifs. After application, Merlin PTFA inferred the genome scale network, which contained around 7,000 ages across these 10,000 genes. Once the network was inferred, we proceeded to analyze the network, and we started with the module analysis, where Merlin PTFA also identifies gene modules where genes with similar regulatory connectivities and similar gene expressions were grouped into the same module. For example, here we see module number 5395, which highly overlaps with the putative module of hypoxia or low oxygen response condition in the fungus. It usually happens when the fungus is trying to grow itself in a diseased lung. So we see that this predicted module recapitulates the putative module for the hypoxia response condition. Then we performed a Steiner tree analysis where we identified the Steiner nodes marked in blue to connect the modules of our interest, which in this case these three modules which produces secondary metabolites. They are molecules which are very crucial for the virulence of this fungus. And we see the first gene underlined here seems to be a module specific regulator of gliotoxin production, whereas the second gene we hypothesize that it might be a global regulator for all these three modules. So we knocked down both of these regulators and we observed that there were significant reductions in the production of gliotoxin and fumaquinazolin and also the other secondary metabolites not shown here when we knock down these two particular regulators. Subsequently, we performed a graph diffusion study where we started with an initial list of genes of our interest in this case the genes associated with lipoketo oligosaccharides or LCOs, which are crucial for the growth of this fungus, and then we assigned initial scores to these genes and propagated or diffused these scores across the inferred edges to see that which other genes, not in the list, receive a very high score. If a gene, for example this particular gene ATFA, has received a very high score, that means it is very well connected to all our initial genes. So we hypothesize that ATFA might be a master regulator of the LCOs, of the putative LCOs related genes. To test that, we produced the ATFA knockout strain as well as ATFA overexpression strain, and we saw that when ATFA is knocked out, then the fungus branches very fast, whereas when the ATFA is overexpressed, we do not observe any branching at all. Finally, to perform all this module analysis and different network based studies, we developed this novel web application known as Merlin Viz, which you can access using this QR code. You may go there and play around with our inferred network. So in summary, we inferred a genome scale gene regulatory network of Aspergillus fumigatus, and we analyzed it to come up with some biological hypotheses, novel regulators and novel pathways, and we also experimentally validated some of them. That is all for now. Thank you for listening, and if you have any question or suggestions, please let us know, and till then, have a great time.